Hi everybody, I'm Karina White and today I'm going to be talking to you about my PhD dissertation research. So in 1936 my grandmother was born and her life expectancy at that time was about 60 years old. And then 50 years later I was born and my life expectancy is 25% longer than hers was. And so it's incredible to me how in a relatively short period of time technology has increased the life expectancies of people and allow us to live long enough to see our grandchildren, our great-grandchildren, to travel, to explore our hobbies more deeply than we're able to when we're working. And I think that this is really a gift for so many people. But with this extended life expectancy, there's some not great things that come along with it. And this is one such thing is age-related degenerative diseases. And so the prevalence of these degenerative diseases has been increasing with increased life expectancy. And so a degenerative disease develops over time and gets worse and can sometimes become such a hindrance that it really limits the independence of the person who's afflicted with it. So, for example, many of you might be familiar with osteoarthritis, which can develop slowly with pain or inflammation in the joints and affect the function of those joints. And eventually, the person might lose some level of independence because of it. Now, one of these diseases is called dry age-related macular degeneration. And in this disease, you lose your central vision. So as you can imagine, if you are used to seeing a picture like this, to seeing your grandchildren look like that, when you begin to lose your vision, your central vision, and your vision becomes to look like that can really be quite traumatizing. And it can really affect your ability to do a lot on your own. And unfortunately, right now, there are currently no therapies that effectively halt or restore a patient's vision once it starts to be lost to this disease. However, there are many approaches being taken to treat it. So this is where my research comes in, and with this research, I'm really seeking to provide hope and progress towards treating this disease and maintaining that ever-important independence of an aged population. So today I'm going to teach you a little bit about the structure of the retina, and I'm going to talk about how my research is seeking to treat degenerative disease that steals many people's independence. Now, the retina sits in the back of the eye, and you would notice the disease is called age-related macular degeneration. The macula is particularly in the center of the back of the eye. So the retina is referred to as this whole area, and the macula is this central region, part of the retina. And so looking more closely at the retinal structure, we have neural cells here. So that's one part of the retina, the neural cells. And those neural cells are what absorbs the light signal and what turns it into vision. And then we have what's called the blood retinal barrier. And the blood retinal barrier is composed of the Bruch's membrane and the retinal pigment epithelium. And it sits all the way at the back of the eye. So it would be lining like this white part. It's lining that white part there. And its main role is to provide support for the neural retina, to keep the neural retinal cells alive so that they can react with the light signal and produce vision. So like I was saying, its key function is to support the neural retina, and it does this by providing physical support. So you can see down here the Bruch's membrane is this little brown region. The retinal pigment epithelium, or RPE, are these gray cells, and you can see those little protrusions that are coming up from the gray cells and how the neural retinal cells sit right in them. So it's providing physical support, which is important. The Bruch's membrane and the RPE also regulate transport, they remove waste, and it absorbs excess light that may come in through the front of the eye. So the retinal pigment epithelium is it's a cell monolayer. So it's a single layer of cells that sit very closely together, and they sit right on top of the Bruch's membrane. And the Bruch's membrane is just that. It's just a support membrane. It's flexible, and it can adjust to different movements. So when you move your eye, your Bruch's membrane is moving a little bit, it provides physical support to the RP, which then provides physical support to the neural cells. However, in aging, the Bruch's membrane becomes stiffer. And so, like I was saying, it can adjust, and it can flex, and it can bend. But as you get older, it can't do that as well. And similarly, the RPE cells don't function as efficiently when you get older. And those two things themselves, they're not the disease. Those are just natural things that occur with aging. It occurs all over your body. However, in age-related macular degeneration, there's cell death. And the cells that die are these neural cells. So you have these two things happening, the Bruch's membrane getting stiffer, the RP cells are losing their function, and you have cell death. And so while these two things naturally occur in aging, they seem to be an initiating step in the age-related macular degeneration disease. As in, these two things happen, which could cause this cell death, or if not cause, contribute to it. So because of this, because these have become, they act as like maybe an initiator, these two things have become a very attractive target for therapy. So if you have a dysfunctional cell in your body, a lot of people might think, hey, can we implant other cells? Can we implant healthy cells to replace these disease cells? How about an implant? And so can we do that? Can we just implant healthy RPE cells? And the answer to that is yes, we can. And a lot of people have been trying this. It's in clinical trials. It's gone through many stages of studies with animals, as well as just studies in the lab. And so we're able to take cells, put them in a liquid suspension, and then just inject them into the eye with a needle. And it's promising, but what we've seen so far indicates that it is a short-term solution. It doesn't last very long, the effects of doing that. And so while it's promising, we're not quite done yet. We think that there could be a better solution in the long term. And so like I was saying before, to remind you, the key functions of this Bruch's membrane RPE blood retinal barrier are to provide physical support, regulate transport, remove waste, absorb excess light. And so when we inject these cells into the eye, what happens is they kind of just form a clump over here. And they form this clump, and then they release proteins and signaling molecules and things that protect the disease dysfunctional cells. So it slows it down in that way, which is great. I mean, slowing it down is definitely a step in the right direction. But what we're missing from this treatment is we don't have this architecture, right? We don't have that single cell monolayer. We don't have that flexible Bruch's membrane that you have in a healthy retina. And so that's a problem. And so this is where my research comes in, which is to recreate the natural architecture and healthy function of the retina. And we seek to do that through implanting RP cells on top of a scaffold. So if you think of the word scaffold, you might be familiar with like scaffolding on a building that helps provide its structure. It's exactly the same concept. We want to provide a level of structure for the RP cells when we implant them so that they're implanted in the proper monolayer architecture. And so our approach in doing this is to first mimic a healthy Bruch's membrane stiffness. So a healthy Bruch's membrane is flexible and able to bend and twist and react to stimuli, whereas the aged or diseased Bruch's membrane isn't able to do that. And so we can do that in the lab. We can choose different materials to make this scaffold out of. We can use different processes to make these scaffolds, and they can all affect the stiffness. And so we can make scaffolds that go from not stiff, which might be like Jello, to very stiff, which if you think of a plastic like a plastic desk or a plastic chair or a pen, plastic from a pen is very stiff. So we can make these scaffolds of very differing stiffnesses, and we can grow the cells on them, and we can see what the cells like more, what more cells stick to, what more cells live longest on. If they are, we do various testing to make sure the cells are happy on those scaffolds. And then our second approach is to promote this functional RP cell by adding a biological cue to the surface of the cells, of the scaffold, So we take naturally occurring proteins and molecules in the body that keep RP cells happy, and we put them on the surface of the scaffold. So then you have scaffold, you have this like decoration, which is the biological cue, and then you put the cells on top of it. And we're trying to make the cells as happy as possible, as functional as possible, and we're keeping them in that native architecture. So just to summarize a little bit is when you have age-related macular degeneration, you have a central vision loss. And what happens is you lose cell function and there's cell death, there's loss of vision, and then there will be a loss of independence for the people afflicted with this disease. And through our approach of using a scaffold with biological cues and RP cells as an implant to replace the dysfunctional cells, we are looking to create a long-term solution, a longer-term solution than what's already out there in just the cell injections. And so we look to replace dysfunctional cells with healthy cells and form a proper architecture. And we believe that through these two things, we'll be able to create a long-term treatment for age-related macular degeneration. So with that, I'll take any questions that you might have about my research or about the disease. Thank you.